Good afternoon, everyone. Welcome to the Wednesday afternoon lecture series. I'm Viviana Perez. I'm the Director of Division of Aging Biology at NIA. It's an honor and pleasure to introduce Dr. Geroscience to today's lectures. Before I introduce her properly, I want to say two announcements. The first one is that the code for the continuing medical education credits is 57685. I will repeat, 57685. And the second one is that for those that are watching remotely, you can submit your question to the speaker anytime during the lecture. Simply click the button that says Send Live Feedback, which is below your videocast window. Type in your message, and we will relay the speaker your question at the end of the lecture. Okay, let's go back to the introduction. So Dr. Su is a professor in the Charles and Marie Robertson Professor in the departments of obstetrics and gynecology, and also in the Department of Genetics and Development. She serves currently as the director of the Reproductive Aging Program at Columbia University Medical Center in New York. Dr. Su is an expert in the field of epigenetics and genetics. Her research has lied in the identification of epigenomic sequence variants linked to the risk of age-related diseases, as well as factors contributing to the exceptional resilience against such diseases, for example, those in centenarians. Her research also examined the cellular response to DNA damage in the context of aging and disease, the role of microRNA and not-coding variants in human aging and longevity, and the enhancer of an epigenetic codes associated with cellular senescence. Her contribution had been recognized widely through several organizations. She received a Glenn Foundation Award, and currently she's the member of the Academy of Health and Lifespan Research. In addition to all her research, she also has organized numerous international symposia, served in numerous editorial boards, and has more than 100 publications. Today we're very lucky because she's going to talk about her latest discovery in science. She is looking the role of aging in ovarian aging. This is a topic that has been recently very interesting in the field of aging because before we didn't hear about this. Before 5 years ago, we never heard of it. But most recently, it's gained really importance because the community have acknowledged that the sex difference play a critical role in the aging process. So the title for her lecture today is Mechanism of Ovarian Aging, Target for Geroprotection in Women. Please welcome Dr. Geroscience.
Thank you so much, Viviana, for that very kind introduction. You hear me okay? Yeah. I'd like to begin by thanking the Division of Aging Biology, Drs. Fei Wang, Stacy Carrington-Lawrence, and Viviana Perez for inviting me to visit and to give a lecture at NIH today. I've been enjoying it very much. I often say that I grew up with Division of Aging Biology since I received my first R01 in 2004 as a junior faculty. And I've been really very grateful to National Institute on Aging and NIH for the support and guidance that really literally shaped and defined my career. So thank you so very much. So as Viviana introduced, my lab has been using human genetics and functional genomics approach to understand the fundamental mechanisms of aging and longevity. And today I'd like to share our very recent work on mechanisms of human ovarian aging. And I hope by the end of my talk, I will convince you that by targeting ovarian aging, we may be able to delay aging in the ovary, thereby making women a little bit healthier. So it's a concept known as geroprotection. I'll use my mouse. Okay. All right, so this is just a quick disclosure. So, I mean, this audience is really well versed in aging biology, but just to frame my talk, I'd like to start with this simple question: why study aging? So we know that aging is by far the single largest risk factor for disease, and shown here is the incidence rate as a function of age, and you see that incidence of all chronic diseases increased dramatically from middle age onwards, both men and women. So for example, just simply getting old from age 50 to 75 increased risk for Alzheimer's disease by 100-fold, which really dwarf tenfold increase in risk conferred by all known risk factors combined, including ApoE genotype, being a female, hypertension, smoking, physical inactivity, and diabetes. And this trend stays true for almost all chronic diseases, different types of cancer, kidney disease, cardiovascular disease, and you name it. So for those of us who study aging biology, propose that since aging is driver of these major chronic diseases, by targeting fundamental mechanisms of aging, we'll be able to delay the onset and progression of many, if not all, age-related chronic diseases. And this Geroscience hypothesis has been formulated based on many decades of very elegant work done in model organisms. So by now we learned that aging is not just simple wear and tear. It's governed by or controlled by underlying biology, regulated by genes and pathways. And when by targeting this evolutionarily conserved genes and pathway, we can extend a lifespan and healthspan in these preclinical models. And by now we know a lot about aging biology that drives aging processes, from genomic instability to nutrient sensing signaling dysregulation, mitochondrial dysfunction, and cellular senescence, and so on. And the drug that target this fundamental process of aging, thereby extend lifespan, healthspan, are collectively called geroprotectors. My lab has been focused on translating these discoveries, breakthrough discoveries made in model organisms to humans by asking questions: is this the same conserved pathway and genes are relevant to human aging and longevity? But more recently, thanks to this enormously large human data set, we can do reverse translational geroscience approach. So humans, by far the best phenotyped species when it comes to aging-related disease and conditions. And now there's an explosive data set, large-scale data set from human genetics to multiomics to electronic health records. So we can really use human as a discovery platform to generate hypotheses and test that in human cells and model organisms. So now, why studying aging in women? As Viviana introduced, women's health has received limited attention. So historically, the biomedical research has been focused on male biology. So study subjects, including animals, are mainly male, to the point where in 2016, NIH mandated the inclusion of both sexes in all research under this sex as biological variable policy. And of course, the funding has been lagged behind. So as a consequence, the knowledge about how female system works and age is limited. And this knowledge gap has really practical negative clinical implications, right? So oftentimes, women are misdiagnosed or diagnosed later, or they respond less effectively to treatment, and they suffer more adverse drug side effects. Luckily, things are changing, and there's more awareness and public attention calling for change, and thankfully, funding too. So just last year, NIH issued special notice encouraging research on women's health. So exciting. So finally, why are we studying aging in the ovary? As I will discuss, ovarian aging does not simply influence reproduction, but it affects the whole body, thereby influencing overall health in women. So first of all, ovary is one of the very first organ to age in the human body. So aging starts in their mid-30s with a rather dramatic decline in function, which completely stops with menopause around age 51. Male reproductive function declines as well, but they are rather gradual. So reproductive longevity or reproductive lifespan is much shorter in women compared to men. And as you know that in the last couple of hundred years, humans' life expectancy has increased dramatically. But the age and natural menopause has remained largely unchanged. What that means is that more and more women live ever greater proportion of their lives in post-menopausal state. And we know this is bad because menopause coincides with very deleterious health outcome in cognitive, immune, bone, and cardiovascular system. And in fact, as measured by epigenetic age, menopause accelerated aging. And there's a genetic component in this menopause-induced accelerated aging. And consistent with this genetic link, women who undergo menopause later tend to live longer than women who undergo menopause earlier. So actually, family with history of early menopause has 6 times higher risk for earlier menopause. And interestingly, brothers of women with later menopause have a pro-longevity benefit compared to brothers of women with earlier menopause. So this suggests to us that ovary does much more than reproduction, and it really influences overall health. So what happens if you remove ovary? Ovaryectomy or oophorectomy increases risk for not just for single disease such as dementia, metabolic syndrome, cardiovascular disease, but it increases risk for multimorbidity and all-cause mortality. Again, indicating that ovary does much more than reproduction. And in mice, simply replacing ovary from old mice with ovary from young mice is enough to extend the remaining lifespan and health span by, in part, improving insulin sensitivity and reducing inflammation. So from the eye of this geroscientist, it was clear to us that ovarian aging can be targeted for geroprotection in women. So what's the underlying biology of ovarian aging that we can target? Unfortunately, as Viviana said, we know very little about basic biology of ovarian aging. Interestingly, what's been well documented, at least in humans, is this enormous heterogeneity in ovarian aging. So shown here is age and natural menopause in UK Biobank, its distribution. And as you can appreciate that there's enormous variability, right? Like many phenotypes in aging. Some women undergo menopause in their 30s, but some women dip in late 50s. So using this variability in age and natural menopause, which, by the way, has been used as a proxy for rate of aging in the ovary, you can perform genome-wide association studies. So one of the largest study involves more than 200,000 women, identifying 290 loci that's associated with menopause timing. And when we look at this variant, 94% of them occur in the non-coding region of the genome, indicating that it's not changing through the changing protein coding sequences, thereby protein function. If it does anything, it does it through regulatory changes, okay, which gives another challenge because a human genome has very extensive linkage equilibrium. So in the locus, you find many, if not 100, of variants that's associated with menopause timing. And regulatory elements such as enhancers and promoter, they tend to interact in a disturbed way so that sometimes skipping many genes in between. So to identify real causal genes that are under influence this regulatory variant is a priori unknown. And regulatory elements, they have cell type specific function. So to identify causal cell types that are under influence of this causal variant and genes are unknown, let alone the mechanisms. So to address this challenge, we and others have been integrating human genetic data from genome-wide association studies to epigenome and epigenetic data set that's ever increasing nowadays, and to map them in regulatory element in different cell types to identify changes in transcription factor binding and look at the association with expression changes. It's called EQTL analysis, and of course look for chromatin interaction to predict potentially causal variants as well as causal genes. And CATLAS, which is a single cell ataxic analysis to look for chromatin accessibility, there are 220 human cell types derived from 30 different human tissues. But until very recently, ovary data wasn't available, which is really unfortunate because ovary is a very complex organ that to maintain this ovarian function. And there are different cell types. So for example, theca cells and granulosa cells are ovary-specific cell type. They are main components of follicles that surround oocyte, that promote oocyte growth and maturation. And of course, there are other cell types such as stroma cells, epithelial cells, smooth muscle cells, endothelial cells of both lymphatic and blood origin, and immune cells. So to address this post-GWAS challenge, as well as to understand the basic biology of ovarian aging, Chen Jin, a postdoc in my lab, decided to take more unbiased, comprehensive, systemic approach by taking single-nucleus, multi-omic approach. So Chen took ovaries from young women in their 20s and reproductively old women in their 50s. So we are talking about aging from 20 to 50, and performed single-nuc RNA-seq for transcriptome profiles, as well as a single-nuc ataxic for regulatory landscape change. And we were happy to detect all major cell types in the ovary. So with this data, now you can integrate the menopause GWAS and map variants. And then we found that the menopause timing associated variants, they occur in regulatory elements that are active in not just one cell type, but across many cell types, if not all cell types in the ovary. Okay? So at the gene at the variant level, they have rather global impact. When you look at the gene level, so predicted target genes, that's in general, it's nearest gene from the variant, we found them are enriched in genes that are expressed in not just one cell type, but almost all cell types in the ovary. You can see it from here. So from stroma cells down, endothelial cells all the way down to thicker cells. Again, indicating this global impact. In comparison, Chen also took GWAS data from polycystic ovary syndrome, which is hormonal disorder often found in reproductively young women. And of course, the hormone is produced in granulosa cells, and you can see that those genes are enriched in granulosa cell specifically. What about ovarian epithelial cancer? So when you take GWAS data from ovarian epithelial cancer, then those genes are enriched in epithelial cells where the cancer is originated. So this data convinced us that menopause timing-associated variants have a global impact on gene regulatory network in the ovary across different cell types. So using this data, now you can start identifying potentially causal variant and causal genes. And I will share with you a couple of examples. We found a variant near HELB locus that are associated with later menopause, and then it occurs in regulatory elements that's active in all these ovarian cell types, from stroma cells, endothelial, theca, granulosa, all the way down to epithelial cells. Okay, so we call it putative causal regulatory variants. When you look at GTEx database, this later menopause associated variants that we identify as a potentially causal is correlated with the reduced expression of HELB. Okay. But when we look at the other tissues in the GTEx database, it was really very surprising that all the tissues that have been profiled, this is 49 tissues, show the same pattern. In other words, ovarian aging associated variants has impact, potential impact, across all tissue type in human body. Okay, so ovary is not even the number one. You can see that, muscle, for example, skeletal muscle, their, magnitude of downregulation is much more than magnitude that you found in ovary. So you can see it from muscle to brain to heart and all the tissues and profiled. And this later menopause associated variant show predicted to bind transcription factors much less compared to non-risk variant. So what I have shown you here is prediction. So you integrate all this available large-scale data, and then you predict potentially causal variants and causal genes. The question is, how do we bring this prediction and association to experimental validation to establish causality? So we've been thinking very hard about the best way to approach this. And Jiping Yang in my lab, another postdoc, he spearheaded this approach. So what Jiping does is he take human pluripotent stem cell, embryonic stem cell, induced pluripotent stem cell, and engineer them with CRISPR so that it carries that causal variant. So everything else is the same except for that single nucleotide. And now you can differentiate these engineered cells to multiple cell types in the ovary, including granulosa cells, endothelial cells, smooth muscle cell, and stroma cells. Now, comparing to isogenic control, you can really confirm the regulatory output. Is this real causal variant that you predict, causal genes, and importantly, it can confirm the directional outcome and also magnitude. In other words, the variant, does it make gene expression less or more. And of course, from different cell types, you can confirm the cell type specificity and really drill down the mechanisms, including transcription factors, and mediate this change. I'll give you one example. Remember the HELB gene, that later menopause associated variant is predicted to show a reduced expression of HELB gene in multiple tissue type, if not all tissue type that has been profiled. So in this case, for the regulatory variant, the best approach is to take heterozygous cells and to perform allele-specific expression analysis. In this case, you have to find coding variants as a proxy for regulatory activity. So when you look at the relative abundance of transcript that carry these proxy markers, you can really deduce the regulatory capacity. So this is control experiment. When you take genomic DNA and perform allele-specific PCR, of course, half and half. But when you take cDNA from differentiated cells, then you see that later menopause associated variant, T allele, cause reduced expression of a HELB gene. Okay, so this is now confirmation of the prediction. And across all cell types that we have tested, again, showing global impact. We are also lucky to get primary granulosa cells from IVF clinic. This is Columbia Fertility Center. We could get 4 cells, a heterozygote carrier of this variant, perform allele-specific expression analysis. Again, T allele is causing reduced expression of HELB. Okay. HELB is a DNA helicase, and it's a inhibitor of homologous recombination, which is error-prone way of repairing double-strand break. And this is also involved in meiotic recombination. So our study demonstrating that later menopause associated variant reduce expression of HELB gene suggests to us that HELB gene deficiency may improve ovarian function, or improve ovarian reserve and reproductive lifespan. So to test that hypothesis, we have generated HELB knockout mice in collaboration with Matt Youssef-Zadeh at Columbia. So we are going to test if this HELB deficiency, as we predict, improve reproductive lifespan and ovarian reserve. And also we are going to test if it has any impact on healthspan in vivo. And this is going to be spearheaded by Shweta Dipali, another postdoc in the lab. Another locus of interest is the famous BRCA1 locus. So this was detected already more than 10 years ago with much less individuals. I think this is like 7,000 women. And it was hailed as a locus associated with reproductive aging in women. Okay? But the question is really BRCA1 the causal gene? As I explained, that the locus, due to linkage equilibrium, has many variants that's associated. In this case, more than 400 variants are associated with later menopause. And then you can see this is very gene-rich region, so it contains many genes, including BRCA1. So how do we know BRCA1 is the causal gene? When Seung-Soo Kim, a postdoc in the lab, looked at GTEx, he found that this variant is correlated with expression changes in MBR2 gene, has nothing to do with BRCA1 or any other genes in this locus. So the putative causal gene in this locus is MBR2, not BRCA1. So to confirm this, Jiping performed again allele-specific expression analysis using this coding variant as a surrogate or proxy for regulatory activity that in each gene. So again, control, when you take genomic DNA, you see 50 versus 50 contribution from each allele. But when you take cDNA from different cell types, now it's immediately clear, I mean, you don't even need to do statistics, that MBR2 is the gene that changes, okay? And more interestingly, the later menopause associated allele cause reduction in expression in BRCA2, okay? Sorry, MBR2. So this is now causal genes in the locus, BRCA1 locus. What about causal variant? So we took 400 variants that linked in this region and mapped them in regulatory elements that are active in ovarian cell type through our single-nucleus ATAC-seq data. And then we found 5 variants, potential regulatory variants in ovary cell types. Okay, so how do we test that? So Jiping in the lab decided to use CRISPR perturbation, so CRISPR-I screen, by generating 2 guide RNA for each potential causal variant, and did specific allele-specific expression analysis after the CRISPR-I screen, and then he found this variant in the distal locus between VAT1 and RND2 gene is the causal variants that dysregulate MBR2 expression. Okay, so through in silico prediction, we identify candidate causal genes and variants, and then through this stem cell differentiation paradigm and allele-specific expression and CRISPR screen, we pinpoint causal variants. Okay, these are 2 locus. Okay. Oh, I have to say this. So MBR2 is a long non-coding RNA that's primate specific. So mice doesn't have it. So it's really interesting that how these long non-coding variants contribute to modulation of ovarian aging. So graduate student in my lab, Maggie Kane, that's her PhD project. She's working very hard to understand the mechanism by which MBR2 contribute to ovarian aging in human. Yes, so 2 locus, 2 loci. Took us a while. But when we did in silico prediction, we found more than 100 potentially causal variants that are linked to age and natural menopause. How are you going to test it? I mean, are we going to pick, our favorite gene and test those, or are we going to do unbiased screen to identify real interesting targets? So since Jiping decided to perform this high-throughput screen to identify causal genes and causal variants in endogenous genomic context by using a single-cell CRISPR-I method. Okay, so this is a simplified schematic. So you have a causal variant in the middle that sits in the cis regulatory element. There are 6 neighboring genes or cis genes, and how do you know which is your causal variant? By targeting CRISPR-I using DCAS9 repressor, in this case CREB. You see, okay, gene F is downregulated by this CRISPR-I targeting. And by combining this with a single cell, you can really look at the downstream transcriptomic wide network effect. Okay, so to be able to do that, Jiping set up this strong dual repressor system, the CREB and MECP2 linked to DCAS9, design gRNA that can be sequenced by 10x, and then you can analyze expression of the neighboring genes as well as transcriptome-wide changes. So GPing did all 101 variants, and it really worked beautifully. So this is granulosa cell. GPing started with the granulosa cell because this is ovary specific. And you can see that compared to non-targeting control, that you see huge differentially regulated genes, about 70 of them, and many of them show downregulation, as you would predict from perturbation. And these are the target genes. The size of the letter indicate the frequency of detection, and you can appreciate that function of these genes are really not known much. So that means we have a lot to do. And I will just present a couple of examples. It was really gratifying to see HELB, the helicase causal variant. When we target it with CRISPR-I, it downregulates HELB gene. Again, confirming HELB is its target gene. We also found new gene, CPNE1, from rather pleiotropic variants up there. And you can see that when you target that variant, that the CPNE1 expression is dramatically down. Again, this is, it's called COPIN1. Nothing much is known about this gene. It's a phospholipid binding protein involved in membrane trafficking and signal transduction. And relatively few studies been done, and one show at least it's correlated with ovarian cancer. So expression of COPIN1 gene is correlated with bad prognosis and more tumor growth. The reason why we are interested in this gene and variant is when you target this variant, thereby decreasing expression of COPIN1, there's an enormous transcriptome-wide impact. So altogether, by targeting 100 variants, we identify about 700 genes going up and down, and more than half of them, or close to half of them, are due to CPNE1 targeting. And when we look at these differentially expressed genes, they are enriched in the so-called hallmark of aging, including mitochondrial dysfunction, like reactive oxygen species, oxidative phosphorylation, chromosome organization, apoptosis signaling. Okay, so this is going to be another set of a study that we go delve into to understand the underlying mechanism of contribution this gene to aging in ovary in human, in women. Okay, so finally, what about aging transcriptome? I mean, we performed a single-nuc RNA-seq in young versus reproductively old ovary. And how do they change across cell types in the ovary? We identify 3,000 genes going up and down with age. And I'm sure that you can appreciate when you look at this, the directional change is the same across different cell types. So there's coordination. And this was quite striking. And Chen decided to look at, what's the extent of coordination. So he could look at pairwise similarity in terms of changing. And this is ovary, different cell types. And you see, you can appreciate that quite a bit of coordination. In other words, they change in a coordinated manner. And then to see if this is ovary specific or it's a general aging phenomenon, Chen also look at other single cell data from other human tissues during aging processes in the same age, including adipose, blood, marrow, brain, liver, breast, and lung. And you can see compared to ovary, there's little or no coordination. So this transcriptome-wide coordinated changes is striking feature of ovarian aging. Another feature that we thought was interesting is that this aging transcriptome, genes that changes with age in different cell types in the ovary, they're really enriched in the hallmarks of aging as we know it. And this is some of the examples, right? So you can see mitochondrial dysfunction, deregulated nutrient sensing signaling, loss of protein homeostasis, epigenetic alteration, genome instability, cellular senescence, and telomere shortening, and so on, right? And again, you see this coordinated change across different cell type. And what caught out our eyes was really mTOR signaling pathway that's activated in the ovary across multiple cell types. And so using that human tissue data from other tissues that we compared, and look at how specific is this to the aging tissues. And so Chen measured this amplitude index as well as the prevalence ratio across different cell types, and ovary really stood out. So at least in this age, mTOR signaling activation is truly ovary specific. And as you know, mTOR signaling is a major control mechanism of aging. And its inhibitor, rapamycin, is best known geroprotector because regardless which lab tests this drug, it increased lifespan and healthspan in all model organisms that have been tested thus far. So very robust geroprotector. And our data showing that this mTOR signaling activity is up across all cell types in the ovary suggests to us that ovary can be maybe a gerotherapeutic target of rapamycin. And this was also in line with the data from mouse and rodent study in mice. Rapamycin treatment increased ovarian lifespan, fertility, ovarian reserve, even under chemotherapy treatment condition. Okay, so together with our data, we now initiated a clinical trial to test the efficacy of rapamycin to extend female reproductive lifespan in humans. The trial is called Vibrant Study, validating benefits of rapamycin for reproductive aging treatment, in collaboration with Zev Williams, who's director of Columbia Fertility Center. And this is really very exciting. So I'll give you a little bit of introduction to this trial. It's a small cohort, 50 individuals, half placebo, of course. So when the clinical trial site became live, we got emails from 200 women. They really wanted to participate in this trial. But as you can see, the study design that we give a low-dose rapamycin, which is 5 milligram per week. This is the dose that people take off-label for geroprotective effects. And then we follow additional 9 months. But during this treatment, they have to come to the clinic every week. So it's really limited to the greater New York area for that reasons. We recruited everyone, and we are almost at the end of the follow-up study. It's all blinded, so we don't know the results yet. But good news is there's really no obvious side effect or toxicity, and even menstrual cycle is not altered. Okay, it's very safe. And primary outcome of this trial is ovarian reserve. So we are, looking at markers of ovarian reserve. But at the end, we also want to collect multi-omic biomarker panel for potential mechanistic insights. And as you can see, this is a pilot proof of concept study. Now we are planning multi-center trial across the U.S. There are 7 centers who are interested in Vibrant study. So we are quite excited about this trial, which born out of our basic science mechanistic work. But obviously, this human trial has also limitations. For example, we cannot get tissues, including ovary. So we set up mouse Vibrant study to mirror this human trial exactly. And so this is a 2-month rapamycin treatment followed by 2 months. And in this case, we can really do a very thorough, comprehensive analysis, including tissues, ovary, and the liver. I mean, the liver, the reason why we took liver was because Vadim, I know that Vadim gave a talk not long ago. He said liver clog, epigenetic clog, is the most responsive to gerotherapeutic treatment. So we decided to take liver, and we are doing very extensive multi-omic analysis from single cell to epigenetic profiling, spatial transcriptomics, and epigenetic age by DNA methylation. Okay, and this is one of the largest collaborative study that we ever did in the lab. So I'm very excited about mouse rapamycin trial. So with that, I would like to summarize my talk. As I say that ovary does much more than reproduction. It influences not just reproductive health and lifespan, but overall health and lifespan in women. And by integrating human genetic data with epigenome, multi-omic data set, we could predict causal variants and genes and cell type, and we validate using stem cell engineering and differentiation model to establish the causality and use them as a target. And our single-cell transcriptomic analysis clearly show that ovary is a model of accelerated aging. But interestingly, it proceeds through the evolutionarily conserved pathway of aging, what we call hallmarks of aging. And this suggests to us that non-geroprotectors that target hallmarks of aging can be used to delay aging in the ovary. So to test this hypothesis, we have initiated Vibrant study using rapamycin as a proof of principle. But I'm sure there will be many more gerotherapeutic agents that we can test in the ovary. So with that, I'd like to thank people in the lab. I think I acknowledge almost everyone, and also wonderful collaborators. Without them, this work would have not been possible. And of course, funding from NIA. Thank you for your attention.
Thank you. Quite a few questions online, but if anyone has a question here, you know where the mics are. I'll start one off because this just came in with the Vibrant, it's called. What is the age range of the women in your Vibrant study? And this is cute. Did the mouse version correspond with the human age range?
Yes. So the human, we went through many different rounds. Initially, we wanted to target women who had difficulty in fertility, but that was very difficult to recruit. So we decided to stick to normal, healthy women in age range 35 to 45, and mouse age sort of aligned with that age range. Max.
Please.
Thank you. Working, right? So, a great talk, Yusen. It's very interesting. Thank you. So, my question is, reproductive system is very special, right? So it's very, very important for evolution. Right? Ovary, for example, is very protected, but age much faster and cease to function at a specific age range, right? So is that possible? Ovarian aging is programmed instead of, general aging, as the other system, right? If that's the case, right? So geroprotector, identified based on, aging of other organs would not work for the, ovarian aging, right? Can you comment on that?
Yeah, that's a very interesting question. Actually, I was really puzzled by our results. And, I was trained in aging biology,
right.
Programmed aging.
The two candidates you identified for GWAS, are not well-known, aging kind of, genes, right? So that kind of, make me to think, that possibility. Right?
Yeah. So I call up a couple of evolutionary biologists, and, is it really programmed? To me, it looks like it. And, of course, there's no clear answer. But, I think it's possible. There are hypotheses why humans have menopause and ovary has to age so much rapidly, like, grandmother hypothesis and so on. So this is something that I would leave it to evolutionary biologists. But one thing that I would like to say is that, when your aging process is so compressed in rapidly aging organ like ovary, right?
Right.
As if, you see this enormous coordination. As if there's a signal coming saying that let's age now. And then you go through very rapidly, right? So I think, when we are talking about translation of gerotherapeutics, we don't have the good way forward, So how do we get there? So I think the ovary can be a good surrogate. So if something works, like rapamycin, I think we can really use ovary as a very rapid test system to test gerotherapeutics.
All right, thank you.
Thank you.
Yes. So it's really fascinating studies, especially the connection link between the SNP to the causing the gene expressions. So my question is simple. One or two questions. So one is probably the regulating gene itself is not specifically expressed in the ovary, probably like the HEL-B or the NBR1, probably expressed in other tissues in the humans. So just one question is the protein that you recently identified is clustered like the HEL-B is like the gene DNA repair systems, or these kinds of any clusters to the identified gene is regulated into some pathways? That is my first question. And the second question is just linked to the just previous mentions that the genes probably not just expressed in the ovary, but also other the tissues. So these kinds of the other, like the liver or the muscle, like the gene expression change is also kinds of beneficial for the fitness of that specific tissues rather than the ovaries.
Yeah, two very important questions. So the first question, so when this menopause GWAS came out, it was a big surprise because when we look at the potential causal genes, most linearist gene, they are really significantly enriched in DNA damage response and repair, which I personally have never seen. This is the only trait that so dramatically show enrichment of the major genome stability mechanism, right? So I think, but they're not clustered. They're all over the genome, right? So somehow there's some selection going on during aging processes in the ovary that's sort of represented in the GWAS. That's number one. Number two, about the implication.
Yes, the not just only for the ovary, but also other kinds of the tissues in the human might be affected by the change in the gene expressions.
So I didn't mention, I forgot. Thank you for bringing that up. So we started with the granulosa cells, the CRISPR ice screen, and we identify target genes and variants. But we are now doing all other cell types in the ovary. That differentiation protocol is available, right? So we expect to see the global variants as well as cell type specific variant and genes. It must be, right? So this is something that we try to tease that out. I mean, so all those predictions through the integration analysis, again, it's a prediction and association. We really don't know if this is really happening in the ovarian cell type. That's something that we are going after. Yeah. So my prediction, yes, you will have global influence, but we'll also find some cell type specific variants, especially follicular cells like granulosa cells.
Thank you.
Thank you.
Eugene, very nice talk. I have two questions. The first one is call my attention the beauty synchronization that you have across all the cell type from the ovary. That's very peculiar because usually when you want to test the aging of a tissue, you usually find a heterogeneity, different markers. Have you checked other aging markers in these cell types? And have something to comment about why do you think you have this synchrony among the cells?
Yeah, that's very interesting question. So again, as I said to Meg's question, I mean, it just as if something is very compressed and the signal makes, age very fast. We don't know. Some other markers that we look is putative cellular senescence markers. In our system, we saw P21 expression level increased quite significantly with age in this setting, and same across multiple cell types, right? So some setting, we see this synchrony. Clearly, senescence is one of them. And I think there's more to be discovered.
Okay. My second question is for the girls that are listening to your lecture. How early is early menopause?
How early is perimenopause?
Early.
Early menopause. Oh, yeah. I think the age 40 is extreme early menopause, 45 early, and then 50, 51 is normal range.
Okay, let me sneak in one question from online. Great talk, explanation point. In the underlying DNA sequences harboring the ovary aging-associated variants, have you found an enrichment of certain TF binding motifs? These TFs may be candidates for mediating the change of the causal gene expression.
We did.
Okay.
Yes, it's published. It's in our Nature Aging paper, just came out. Yes, CEBP delta is, we think it's a master regulator of ovarian aging across different cell types. So that's quite striking, actually. I mean, due to the interest of time, I didn't talk about it. Yes, so the audience can find that information in the paper.
Okay.
It's very interesting. Yeah.
Good.
Yeah. So my question is on the iPS cells. You differentiate into ovarian cell types. So I'm trying to channel Tiziana here. Right? There's a big NIH Common Fund initiative lasted many years already on microphysiology systems. So I'm thinking for developing, say, using ovary as a testing platform for gerotherapeutics. Wouldn't it be nice if you could build some small microphysiology systems for...
Oh, I'm all for it.
Yes.
Tiziana, please sign me in. Yeah, I can we can provide ovarian cell types. I mean, it took us a while to establish differentiation because, but yes, please.
Yes,
We are also, there's a postdoc from Francesca Duncan's lab. She joined my lab, and she developed ovary organoids. So we are now trying to develop human ovarian organoids by adding different cell types. So that would be really cool, yes.
Okay, so that's actually was my question. So are you working on the organoids, or are you thinking to literally engineering three-dimensional tissue models?
That was a postdoctoral award application. So we are using organoids first to screen known geroprotectors, and then prioritize, and then go to real ovary.
All right.
That was the whole plan.
Yeah.
Mouse
Thank you.
study.
Thank you.
Thank you.
Just a question about the rapamycin study.
Yes.
Presumably in preparation, you did survey the published clinical literature on use of rapamycin. Substantial. Was there sufficient data there to be able to look to see from the natural history, medical records of those involved in those studies, whether there was any hint or clue of effects on menopause?
Yes. We tried very hard to find such information, but thus far, the rapamycin takers are older individuals, mostly 60 plus. We could find one younger woman, 50. So we really couldn't do it. So this is really very unique in the sense that younger women are in trial. Yeah. And we were very happy that it's okay. I mean, no one suffered from anything, even mouth sore. Maybe because young people are more resilient.
Okay, maybe I could sneak in one more question.
Sure.
Holding up? Okay, because there's lots of them, but this is a quick to say. What happens when your ovaries are surgically removed? How does that affect anything?
Yeah. I had a slide. Maybe the person missed it. So if you surgically remove ovary, so this is surgically induced menopause. I mean, it's not good. They have shorter lifespan, and they have a higher risk for age-related diseases. So nowadays, at the time of hysterectomy, they try to save ovary because if you conserve ovary, they tend to live longer and suffer less with age-related disease compared to completely ovariectomized women. So it's well established epidemiologically. Yeah.
Okay, thanks. So, okay, one more question up there. Good.
Hi, nice talk. So my question is related to the previous question. So for those ones, as you mentioned, that those ones who have the ovary removal, they have a lower reduced mortality, right? I mean, increased mortality. So I'm wondering, does treatment with the geroprotectors help reduce their mortality?
Yeah, no one have ever tested, but that's a very good way to start, right? So we've been thinking a lot about how to expand our very vibrant study. So now we start with the young, healthy women, but women with difficulty in getting pregnant or premature ovarian insufficiency. So we will have immediate benefit if it works, right? So we've been talking about this, like, POI patient, POF patients. Yeah. Thank you.
Yes. Do you want one more comment? Okay. I'm sure there's an obvious answer to this, but the negative effects of surgical oophorectomy presumably persist despite best efforts at hormone replacement.
Yes.
So do you have any idea of what the what speculation there is about the systemic effects beyond the measurable hormone changes? Yeah.
I can tell you two stories. Number one, based on our study, across all the cell types in the ovary, it's not just follicles that produce hormone, but stroma cells, endothelial cells, everybody shows the same phenotype. In other words, the ovary interacts with the oocyte. The germ cells interact with the soma, and they influence each other. So you can actually rejuvenate old oocyte with younger, follicular environment. That study has been done and published in Nature Aging not long ago. This is a Singaporean group. So they reconstitute old oocyte with the young follicles, and then they show that oocyte is rejuvenated. It started to show functions, right? So that's number one. Number two, in mice, so I explained this transplantation experiment. So if you replace old ovary with young ovary, it's beneficial. So in this experiment, although the sample size was really too little, so I cannot make conclusion of it, but the effect was dramatic. So what they did was, before transplantation, they removed follicles that produce hormone. Okay, there's a chemical, VCD, that removes follicles. So now you have a young ovary without hormone. That was enough to extend lifespan. I mean, looks even better. In other words, the somatic compartment of ovary have pro-longevity effects. Okay, so when we look at our data set from single cell, I really think that soma does a lot to the health of the ovary.
Very good. I'm just going to repeat our CME code, 57685. Remind people there's a reception just outside the doors here, and maybe thank our speaker one more time.
Thank you so much.